Coming up ahead in this episode of Xtalks Spotlight.
As a technology itself, bispecifics are really growing. So they're kind of, becoming much more popular in terms of the overall concept that people are exploring. And actually there are hundreds of bispecifics in preclinical or clinical trials at the moment, with the main area of focus being oncology. So it's a really growing area.
There's not really a one-size-fits-all approach for a bispecific. Screening things like the avidity of your bispecific, the spatial arrangement of the different arms that you're adding together. These things can be quite critical, especially for function, but also for the overall stability of the molecule that you're making.
Hello, and welcome to Xtalks Spotlight, illuminating insights from subject matter experts and industry thought leaders. I'm Sonia Hunt. In this episode, we're exploring a critical question. From concept to clinic, are we making the right first moves in bispecific development?
Bispecific antibodies are designed to bind to two different targets, offering new ways to approach complex diseases. Bringing them together in a single molecule isn't so simple, however, as it's often a balance between format, function, and feasibility. And making the right choices early on can shape how far a project goes. In this Xtalks Spotlight edition, I spoke with Arron Hearn and Thomas Cornell from Abzena's protein engineering team. Together, we explored the strategies they use to guide bispecific development, from selecting the right format to spotting challenges before they slow progress. Whether you're weighing bispecifics for your pipeline or already mid-design, this discussion offers timely insights into how early choices shape downstream success. Thank you for taking the time for the Spotlight interview, Arron and Thomas.
Thank you for the introduction and looking forward to the discussion.
Hi, I'm very much looking forward to it.
So Arron, I'll start with you. In your experience, are more teams you work with at Abzena beginning to explore bispecifics?
Short, the answer is yes. So it really opens up the possibilities to look at other mechanisms of action that are limited by traditional monospecific antibodies. So it allows you really then to look at, potential unmet medical needs, which we wouldn't traditionally have been able to look at and access. So for bispecifics, there are three main mechanisms of action that is possible. So the bispecific can target two epitopes within the same protein. Those epitopes may be on separate proteins and be able to bring them together. So there's a really good example in the clinic of Hemlibra, which brings two clotting factors together for hemophilia A. And so then that allows the patient to be able to have blood clotting. And so that's a great example of a bispecific in the clinic, which, is really being able to function and have a positive impact in terms of patient health. And then there's another example where the bispecific can bring two cells together. So for Blincyto, so that's a T cell engager. So that enables a cancer cell and a T cell to come together and enable cancer cell killing. So again, another great example of being able to use bispecifics to bring two things together and have a very positive outcome in terms of unmet needs and oncology.
And Thomas, could you share what bispecific formats Abzena typically supports in early development?
That's a really good question. I mean, there are many formats out there, and one of the interesting things about bispecifics is the innovation that's happening around them and the amount of technologies and sort of different abilities to create them. I would say that there's probably sort of two main groups of clients that we deal with. We do not have a specific technology that Abzena uses. And so we're quite agnostic to different technologies and techniques that are out there to be used, and we can work with a variety of them. Clients tend to come with two main requirements. One is either to have a IgG-like bispecific molecule, so that would maintain the traditional kind of shape size of an IgG antibody, or people who are more willing to explore different shapes and sizes as well. If you wanted to maintain an IgG-like bispecific, then these tend to be IP covered technologies that are out there. So things like Fab arm exchange, DuoBodies, cross mabs, technologies like that are quite common to be used for such examples. For non-IgG like designs, again, this sort of tends to move you away from IP and restrictions that you might face there. And that tends to require reformatting one of the arms in order to get away from light arm exchange to things like reformatting as a single chain FE, using a VHH or other formats like that, and going for a more asymmetrical based approach. And so with any of those technologies, we're able to guide clients through what might be the best technique for them, and to sort of highlight the pros and cons of each way.
Now, building on that, how close are bispecifics to clinical approval, and are there any already on the market?
Making bispecifics is really challenging, but it's very, very doable. If you look at the clinical landscape at the moment, only 17 bispecifics have been approved as of December 2024. Now, most of those are kind of focused on very traditional IgG-like molecules, so they're asymmetrical. So they do look like antibodies in general. So those are built using DuoBody technologies that allows controlled Fab arm exchange to bring two antibodies together. Those may use cross-mab technology, for example, to get over light chain mispairing. And some of them also have common light chains as well. So again, all of these technologies get around the major problem of building bispecifics, which are light chain mispairing. There's also another couple of examples which are in the clinic as well, which are single chain molecules. So those are much smaller in terms of the way that they're constructed. They're effectively a fusion of multiple proteins together in order that you can express them, purify them. And so then there's no complication in terms of expressing multiple different proteins together within the same cell in order to be able to construct the bispecific. As a technology itself, bispecifics are really growing. So they're kind of, becoming much more popular in terms of the overall concept that people are exploring. And actually there are hundreds of bispecifics in preclinical or clinical trials at the moment, with the main area of focus being oncology. So it's a really growing area. And, within time, it will probably overtake monospecific antibodies as kind of the major force of antibodies within the clinic and treating patients.
So now we have covered bispecifics, but let's talk about multispecifics. Just how important are they?
Interestingly, although there are many bispecifics in clinical trials and some have been approved, actually as of now there are no multispecifics that have been approved in the clinic. Although there are some clinical trials going on, those are much less in number compared with the bispecifics. But they are ongoing at the moment, and as you can imagine, there are complications in terms of producing bispecifics. When you start talking about multispecifics, the complexity rises even further, both in terms of production, but also in terms of safety concerns as well. So, those multispecifics are binding multiple targets. And so, there are additional safety concerns, but there's also additional thought that needs to be put into how you're manufacturing them and how you analyze them as well in terms of characterize them during manufacture as well. So when we talk about multispecifics, we're talking about, not only just antibodies per se, but we're starting then to think about additional fusions as well. So, antibodies fused to cytokines, for example, for delivery to tumors. And so, these molecules are starting to get to much more complicated and difficult to produce. And also, when you start to think about multispecifics as well, you start to think about ways that you can improve bispecifics as well. So, generating antibody drug conjugates with bispecifics. So that has an additional payload on the bispecific in order to be able to kill cells, for example. And in addition to kind of drug conjugates, we're starting to see oligoconjugates as well. So oligos that are conjugated to bispecifics in order to be able to deliver those into cells, in order to be able to knock down protein production in those cells as well. So there are many, many different ways of adapting a bispecific or generating multispecifics as well.
And now to wrap things up, Thomas, what would you recommend if you were to talk to someone who wanted to start a bispecific project?
Yeah, there are many considerations, I would say. So there are a lot of complexities and there are a lot of challenges with doing bispecific programs. You cannot simply take the biophysics perhaps of the previous antibodies and dictate directly how well or badly a bispecific might perform. So there are always risks around aggregation. If you are reformatting into single chain FVs, then there are risks of losing affinity, losing function, low yields, for example, things like this. So there are a lot of challenges when it comes to developing and designing bispecific antibodies. I would say that the best approach or the best advice that we can give is to encourage people to try more than one design. So, there's not really a one-size-fits-all approach for a bispecific. So whether that's trialing different technologies, again, whether they are covered by IP or not, might be the best way to sort of give you a broader base with which to try and find the successful design that you want. Screening things like the avidity of your bispecific, the spatial arrangement of the different arms that you're adding together. These things can be quite critical, especially for function, but also for the overall stability of the molecule that you're making. And so designing a smart panel of multiple designs is, I would say, a key requirement to think about when going through a bispecific program. Coupled with that, I think, is that although you can design many bispecifics relatively easily, and again, that's something that we can guide clients with as well, having a very good screening process is critical as well, because although you can design many of them, you need to be able to triage them as quickly and effectively as you can. So getting early binding affinity data, having a very good bioassay system that allows you to pull out the design which is giving you the optimal biological output that you want is also critical. So I think in short, there are many challenges. These things can be overcome, but certainly going for a broader selection of bispecifics to begin with and triaging down is a good approach to take.
Well, thank you very much, Arron and Thomas, for speaking with us today. We really appreciate your time and insights.
Thank you very much. It's been a really interesting talk. Thank you.
Thank you. It's been a pleasure.
We look forward to learning more about how Abzena supports biopharma teams working through the complexities of bispecific design. Thank you all for joining us for this Xtalks Spotlight feature. We hope you enjoyed the discussion.